Wednesday? How about semi-conservative? Remember that term. DNA replication is semi-conservative. And see, here's that 3' on one strand, 5, 3, and 5. So they are anti-parallel. But since replication can only occur in one direction, one strand is continuous. So it's always ahead of the other one. So it's the leading strand. The other is discontinuous in those small pieces known as Okazaki fragments. Therefore, that is the lagging strand. It always lags behind.
DNA replication begins at a specific point in the DNA molecule called the origin of replication site. Initially, the enzyme helicase unwinds and separates a portion of the...
Yeah.
Do you put names or what the lab partners should I have to see that.
Okay. Yeah, I want to know Wednesday if you can, because we want to be able to start putting it together.
Okay. 4 programs. I got it. My spell's not.
Okay.
Sorry.
No, that's okay. But,
Good luck.
But that's what we, because we want to talk about that Wednesday in lab. Okay, and helicase, enzyme that unwinds the helix. Okay, and you'll start with what's called an RNA primer, obvious.
DNA molecule, after which single-strand binding proteins react with and stabilize the separated single-stranded sections of the DNA molecule.
In other words, they keep it apart so we can replicate.
The enzyme complex DNA polymerase engages the separated portion of the molecule and initiates the process of replication. DNA polymerase can only add new DNA nucleotides to a pre-existing chain of nucleotides. Therefore, replication begins as an enzyme called primase assembles an RNA primer at the origin of replication site. The RNA primer consists of a short sequence of RNA nucleotides, complementary to a small initial section of the DNA strand being prepared for replication. DNA polymerase is then able to add DNA nucleotides to the RNA primer and thus begin the process of constructing a new complementary strand of DNA. Later, the RNA primer is enzymatically removed and replaced with an appropriate sequence of DNA nucleotides. Because the two complementary strands of the DNA molecule are oriented in opposite directions, and the DNA polymerase can only accommodate replication in one direction, 2 different mechanisms for copying the strands of DNA are employed. One strand is replicated continuously toward the unwinding, separating portion of the original DNA molecule, while the other strand is replicated discontinuously in the opposite direction with the formation of a series of short DNA segments called Okazaki fragments.
That's the lagging strand.
Each Okazaki fragment requires a separate RNA primer. As the Okazaki fragments are synthesized, the RNA primers are enzymatically replaced with the appropriate DNA nucleotides, and the individual Okazaki fragments are then bonded together into a continuous complementary strand.
DNA ligature binding. If we go back a little bit.
Of a series of short DNA segments called Okazaki fragments. Each Okazaki fragment requires a separate RNA primer.
Right here.
As the Okazaki fragments are synthesized, the RNA primers are enzymatically replaced with the appropriate DNA nucleotides, and the individual Okazaki fragments are then bonded together into a continuous complementary strand.
And that's the DNA ligase that does the binding. Now, does that make sense to everybody for DNA replication? Semi-conservative. It occurs obviously in opposite directions on there, but it's the same direction as far as the bases are concerned. But because they're anti-parallel, it's opposite. And so therefore, one can be continuous, the leading strand, and the other is discontinuous. It's always behind the other one, so it's the lagging strand. And those fragments, those segments, short segments, are those Okazaki fragments in terms of replication of DNA. Now, the other aspect of this, we get into that genetic code. And it specifies the sequence of amino acids in the polypeptide chain, which is what? That primary structure. That's that primary structure. Okay? So each amino acid is represented by one particular triplet on DNA. And its complementary triplet on messenger RNA is known as the codon. The triplet that matches up and is complementary on transfer RNA is the anti-codon. So, again, don't try writing this down straight away, please. We're talking about different things about it. You try to write it down, you're going to miss what I'm saying, and guess what? Then you're going to be like, what in the heck is that? Or some other version thereof that might have expletives that require deletion. We don't want that. So, yeah, the genetic code specifies for the sequence. Well, each amino acid is represented by a particular triplet, which means a group of 3. And that's on the DNA. A complementary triplet on messenger RNA is the codon. Now that codon is going to have a complementary triplet on transfer RNA. Messenger RNA carries the code. Transfer RNA transfers the amino acid to the chain. So it carries the amino acid and the anti-codon. Carries the amino acid and the anti-codon. Transfer RNA, tRNA. Transfer RNA carries the anti-codon that matches up with our messenger RNA and carries the amino acid too. It transfers the amino acid to the chain. Messenger RNA has the codon that carries the message. And the original triplet on DNA, again, because it all encodes for one amino acid. Each transfer RNA has an anticodon associated with one and only one amino acid. Therefore, obviously, the codon on messenger RNA, which is complementary to that, is for one and only one amino acid. Likewise, that triplet on DNA encodes for one and only one amino acid. And the specific sequence of those on DNA tells you what the protein is. It tells you what the protein is. So DNA is in the nucleus, stays in the nucleus. So we got to do what? Send that message out to where proteins are made. And where are proteins made?
Nucleus.
Where are proteins made? Ribosomes. Not in the nucleus. They're not in the nucleus. Remember, ribosomes are made in the nucleolus, yes, but ribosomes are where proteins are made. Ribosomes are for protein synthesis. Remember, free or attached to endoplasmic reticulum, that makes the rough ER. That's why we bothered to study that, because you got to understand some of it. And ribosomes are out in the cytoplasm, so we got to take that message of the DNA and send it out to the ribosome to make the protein. How do we do that? By transcribing it onto messenger RNA, transcription. And that process of going from DNA to messenger RNA to a protein is known as the central dogma. And Transcription is making messenger RNA from the DNA sequence. And RNA molecules are single-stranded. Uracil replaces thymine. They are shorter. There are a couple of other types of RNA, and we're going to talk about them in a few moments before we're done tonight. Messenger RNA, which carries the code, transfer RNA, which transfers the amino acid to the sequence, to the chain, and ribosomal RNA, rRNA, which is part of the ribosome, kind of like scaffolding. Kind of helps provide the structure upon which translation occurs. That's what rRNA does. It helps supply that structure. tRNA transfers the amino acid, and messenger RNA carries the code. So we transcribe DNA to messenger RNA much like a transcriptionist in court. Or anybody ever given a deposition, like in a lawyer's office? And you have a transcriptionist there who transcribes that information. So this is an mRNA transcript when we're done. So that's why it's called transcription. And protein synthesis, again, does involve those two steps. Transcription, taking the DNA onto the messenger RNA. Translation at the ribosomes, out in the cytoplasm now. Transcription occurs in the nucleus, and it transfers the information to that chain of amino acids. And we have that polypeptide chain, that chain of amino acids. When we're done, what do we have? The primary structure of a protein. Yeah, we have the primary structure of a protein. And now, when we transcribe messenger RNA, the section of DNA, it opens up where we need it to. And the RNA nucleotides pair up in an appropriate manner with the DNA. So if it's adenine, what do we get? Uracil. If it's thymine, we get adenine. If it's guanine, we get. If it's cytosine, we get guanine. It's that simple. And RNA polymerase controls the process. And then once the mRNA is made, it goes away. It's going to go out to the ribosome, and the DNA is going to close itself back up into the double helix and stabilize itself. And this is going to talk to you about the stages of transcription and what we get when we're done.
A structural gene is made up of a sequence of bases in a DNA molecule consisting of a coding region with an upstream promoter